thanks very much. I think I hear the microphone. That's good. We have 50 minutes. We have 40 minutes to talk among ourselves, and then 10 minutes to field questions from you. It's actually, you'd think that that's not very much time to talk about modern medicine. Where is the science taking us? But as a radio reporter, 50 minutes—that's a pretty good chunk of time. So we'll see what kind of progress we can make on a whirlwind tour through this. And the first question I'd like to ask is actually to all of you, each of you, which is just sort of stepping back and reflecting that we're in a really, truly remarkable time in biomedical research right now. The technology has come along so amazingly. We have the genome was sequenced more than a decade ago. We have deep sequencing now. You've got all of these knockout mice and so on. You've got just the big data. You've got personalized medicine becoming precision medicine. I mean, it's just amazing, all of this foment. And people are turning and saying, okay, this, on the technological side, the progress is fabulous, but it doesn't seem like the actual results in clinical medicine are keeping pace with the technology. And I'm wondering if each of you would sort of reflect a little bit on what it would take to accelerate that transition from having these fabulous tools to actually making them, putting them to use more rapidly. And I guess we could start. We'll just move down the line. I guess, Ron, would you like to start?
Sure. Thank you very much. I have to say it's a great pleasure to be here in Washington. And actually, I was thinking about your question. Many of you know this very well, but I recently happened across an NSF paper from 2004 talking about the confluence of 4 particular areas, and they named them as nano, bio, info, cogno. And some of you have probably read that paper a long time ago. And when I think about what we see in our company around the world of research in general, and medical research in particular, you see that confluence actually taking place. And I reflected on 2 things. First of all, it's a testament to the forward-thinking nature of research in the United States, that 10 years ago, this trend was identified, and now today, you're seeing the impact of those trends flourishing. My general answer to your questions follows on from that observation, which is it seems to us at Elsevier that we are at the cusp of making that promise a reality. When you see the type of work that's being done, you're moving from the theoretical, the conceptual, to real practical implementation. Some of the comments from the speaker earlier on supported that. Evidence-based medicine, going into the patient, talking to the patient while they are being treated outside of the trials. And so from our perspective at Elsevier, we see the research, we see the clinical impact, and when I talk to customers or when I talk to our own employees who are involved in understanding what's happening with research in clinical medicine, we all are coming to this conclusion that we are right at the cusp right now.
Yeah, and Dr. Gibbons, obviously, it's as part of the NIH, it's your institute's job to bridge that gap, right? How are you thinking about that?
Absolutely. Well, I think it is an exciting time. I reflect for the National Heart, Lung, and Blood Institute on, I think, one of the major success stories in reducing coronary heart disease deaths by over 70% over the last 50 years, in large part driven by investments in biomedical research. So we have classic studies like the Framingham Heart Studies to identify mediators of disease that then led to basic science research, Nobel Prize-winning work of Brown and Goldstein to identify targets that then pathways that were druggable, and public-private partnerships to develop cornerstone therapies like statins. And that took a period of time. What's remarkable is we've basically repeated that cycle just very recently, very quickly, with the new PCSK9 inhibitors, in which, again, genomic science now sequencing people with the tails of distribution of cholesterol can identify a new druggable target. And we already have those that are FDA approved within a rapid period of time. That's the nature of this technology. We're also excited about the big data that's emerging. So in Framingham, people would come into a clinic visit every 2 years. Now when you think about what we can capture about their blood pressure, their heart rate, their sleeping habits, that is now captured by mobile technologies and sensors, we have the capacity now to create data sources that we can then layer on genomic information about those same participants. So at NIH, we now can create data sets that are incredibly rich, that not only involve those classic clinic visits, but actually data that's person-centric, that is derived from the new technologies. And I think the challenge for us is to promote a culture of data sharing where all of that will be available for everyone to mine, whether in the public sphere or in the private sphere, so that those new targets, that new understanding of mediators continues to advance.
Yeah, obviously the Genome Project sort of set the standard for data sharing for sure. Neil, Gina, tell about this from the perspective of a pharmaceutical company. How come you aren't giving us wonder drugs every other week?
Sure. Thank you very much for the opportunity to comment and for the opportunity to be here. I certainly would agree with the point, which is, we're at the cusp of something which is an evolution in the way we manage healthcare, from a research right through to a delivery perspective. I think the one thing that I would say, not even looking at Shire and pharmaceutical or biopharmaceutical companies in general, one of the key things that I think that we would need to do to progress this is to break down silos, is to enable a lot more partnership. I think whether you're talking about working clinically in a hospital setting, or working in an academic setting, or working in a research setting, or working in the pharma setting, or working on the government or the payer or the delivery setting, we've gotten better over the years, partnering, within a certain elastic state with various partners that you must partner with. But broad partnership across the board and a coordinated approach to partnership, I think, is lacking. And I think that's one area, if we really want to truly embrace this next level of precision medicine, or next level of more personalized medicine, I think we're going to have to break down those silos. I notice it a lot, obviously, at Shire. We do a lot of work with rare diseases. So we know that we don't develop all those ideas in-house. We collaborate a lot with external partners who have great ideas. And our area of specialty is about taking those great ideas and hopefully bringing them to a patient ultimately. But you see, or I've certainly seen in the last few years, going back to that breaking the silo and working with other partners, working with regulators in rare disease, working with payers in rare disease, working with other researchers and other companies in rare disease is totally different than my experiences in working in other larger pharma companies, because that silo just has to be broken down in order to have that next level of innovation. So for me, that's definitely one area to think about, is how do we just have policy or ideas from all the different stakeholders involved at this particular cusp to break down those silos and enable us to work better together.
We should circle back and talk a little bit in a few minutes about the role of the federal government, what it could do to help on that. But let me kick the question down the row here to Vince Forlenza. What is your take on this?
Sure. So from the perspective of someone who's been in the medical device and diagnostics industry, I can remember doing funding my first biomarker discovery program back in 1984. I think fundamentally we understand the complexity of the science, and thinking about what's going on in genomics. It's totally different than the approach we were thinking about 20 years ago with identify the single oncogene. We have a tool set now that is so much more powerful. Those tool sets, though, are still very complicated. When one thinks about the translation from the research to actually a clinical diagnostic, for example. And it's every step of the process. I think the exciting thing is that there is a lot of funding going into improving that tool set, but how you take that so that it's a repeatable process with the right sensitivity, number one, so that laboratories can employ it, and then two, get it through the regulatory pathway and get it paid for is really important. And that's where I think the 21st Century Cures work is going on. AdvaMed has been also working with the FDA on this issue as a separate breakthrough pathway. So I think we have to solve those problems first. And then on the back end, when you have the result, the understanding of what that result means in the physician community, because these are going to be complex, as I said earlier, I think you have to solve that from an information and understanding standpoint.
Sure. I may be building on a theme here, and at the risk of saying that I'm Everything's been said, except not everyone has said it yet. I'll be brief. I think the key point...
I can throw you different questions in if you'd like.
For me, I think, first of all, the fact that my primary care physician is talking in these same words means that we've gone past the cusp. We're actually at the point where the dam is starting to break. And what's amazing to me is that this is by an organic nature. Through the silos have all made progress on their own. And to me, the biggest thing we can do to build on the theme of breaking down the silos is to create something that's beyond the organic progress to something that actually has a strategy behind it, something that actually builds towards something, the final vision. And we need that. And that, to me, is what's going to break down.
Well, a lot of biomedicine in this country is focused, certainly, and academia is focused on the individual investigator, the R01 grant, et cetera, and people pursuing really, interesting ideas. But is that the right model? If we're obviously, then it shifts into pharmaceuticals. But is there some need to rethink that model? Do you think, or do you think that that it needs to be? That's still the right way of thinking about it. And
I certainly think that since we fund the R01s, I think that we want to be innovative and adaptive. As we mentioned, we have the capacity now to create data sets that are accessible to more people with more analytical tools than ever before. And so I would agree with an element of that premise that funding sort of mom and pop little elements alone is probably not sufficient. But I would dare say that one of the strengths of that model is to empower individuals to basically follow their nose and to try to solve difficult problems. So for example, if we're thinking about something like gene editing technology that really potentially could transform how we think about curing certain diseases, like in our portfolio, hemoglobinopathy, sickle cell disease, you then have to step back as to how does that tool come about? Well, it's because someone was studying the immune system of E. coli. And there are not a lot of advocacy groups for E. coli. And yet, those tools, those technologies, that understanding was fundamental to creating this tool that then could translate. So I think we need to be careful that we maintain that balance of allowing brilliant scientists to follow their nose and to ask how systems work, as well as this translation, and have that as part of a broad portfolio.
Ron, did you want to add something?
Yeah, let me build on that. I completely agree with the point about the balance. And from our perspective, when we talk to government funders around the world, we hear that question about balance, but it's calibrated a little bit by something which I think is a little bit new and a little bit recent around the need for demonstrating value, demonstrating return. There's a little bit more of a pressure on what benefit does the taxpayer get for this investment, and being able to demonstrate some kind of metric back to the taxpayer that there is a measurable return. So that balance between allowing the individual researchers to follow their noses, which I completely agree with this, how do I measure? And we've been doing quite a lot of work around that sphere, because if you can't convince the taxpayer of the value, then you don't get the funding to follow your nose. So you have to balance these two, and so we spend a lot of time on that. The second point I'd make is around the role of government, aside from funding, around creating frameworks. And that actually was mentioned in the earlier introduction. There are some times when governments need to provide a framework from which industry, commerce, other organizations can work. In the absence of frameworks, it's very difficult to coordinate the different stakeholders effectively to get to the result, to get to the promise that you were describing earlier.
Yeah, and Sudip, I know you have spent time on Capitol Hill and think about science policy issues. Is there some need to change the incentives a little bit to scientists and say, maybe we need you to be in a team and working on a team on a particular project and not worried so much about proving something that will get you your next R01 grant, and I mean the whole structure is in an interesting place right now because it
is. Yeah, the framework is one that has really remained the same from Vannevar Bush forward, right? It's the same framework, and it's been wildly successful. So we have to be very careful when tinkering with something like that.
Indeed,
But I think NIH has already been experimenting with some ways of saying, what are the incentives for a young investigator? Do I want my name on a publication with 100 authors, or do I want my name on a publication with 3? The
And
incentive.
physicists don't mind being on a paper with 100.
They don't,
But
that's
the,
right.
but the biomedical scientists, no,
They
not
do.
so much.
They do, and I think that is something that the culture is slowly changing. The other piece of that is that you have to have both, and so how do you build those cultures both separately and together at the same time? And that's a very challenging problem for NIH and for NSF and for any other funding agency. But I think that the key to that is seeing the problems that can be tackled. So when you think of something like CRISPR or some other technique that's profound that comes from basic research, it's wonderful to have those folks follow their nose. But then it's also wonderful when they can frame it in a way that policymakers can say that's valuable. So I'll give you an example. I used to work for Senator Arlen Specter, and I was very excited. He asked me, what's something great that's happened in biomedical science? And the structure of the ribosome had just come out. So for the science geeks in the room, the ribosome is a really important piece of protein material, protein and RNA material. And he said, okay, why is that important? And I said, well, it's, we're seeing that the RNA world predated the DNA world. And he said, I don't care. And so something I can care about. I said, well, it turns out that you can actually use this information to create drugs. Suddenly, that's where the value of NIH from a $7 billion agency versus a $31 billion agency became apparent to a policymaker who's a lawyer and who has to walk into the next room talking about worries about defense. You have to make it very clear. And that's something that young folks, the new scientists, the younger scientists can start to talk about. And that culture is changing.
Yeah. So,
So maybe this is from a totally different point of view, but as a manufacturer, we are seeing, so this is not directly in terms of NIH or whatnot, but if we look at NGOs and other organizations looking at, for example, the developing world, we're starting to see public-private partnerships being formed in the research area and in the commercialization area from a shared risk perspective. And figuring out both how can they get paid back and under which circumstances, which is a pretty interesting model. So it's enabling you to take more risk. And in addition to that, they're looking at shared value. So where are they going to put the money and how are they going to create these frameworks that we were talking about before? They're starting to build in the societal impacts. Now, I wouldn't say this is standardized. I would say it's all initial work that's happening right now. So maybe there's some analogies here in terms of the pure research side of things.
yeah. So, and speaking of value, I think it's a word that I expect will be said often today, and it's an interesting issue. I think one of the value that people are interested in is not only having more medical advances, but having them remain affordable. And I wonder if you guys would like to talk a little bit about whether technology inevitably makes things more expensive, or are there opportunities with using these technologies actually to make, to get more bang for the buck out of these things, and to have, whether it's diagnostics or treatments or whatever, actually be more affordable. So,
Well,
Neil, do you want to do
I
with
can
that?
certainly start with that.
Yeah.
And it picks up on some of the themes that were highlighted by my colleagues just in the last few minutes. I mean, I think the amount of research and what is discoverable is phenomenal. And I think allowing people to keep pursuing those ideas with all the different platforms that are available is very important. But it goes back to a point that you were raising earlier, is from a society perspective, how valuable are they? So I think it's very important to keep those incremental pieces of research going and going off down different rabbit holes in whatever direction they need to go to enable that innovation moving forward. But what are the important things to think about from a society perspective? And then it gets back to that question, the value, right? If it's a value to society, if it's a value to a certain group, then it should be valuable to a number of groups to come together to think about how we actually can achieve this. And I think that's one of the problems. We see it, every other day in the industry. You come forward with great ideas, with great targets that you've been able to fulfill to hit a small number of patients or maybe a large number of patients, and you get down to that point, the cost to society, the cost to payers, and the incremental change, is it actually certainly valuable to those patients, but in the wider sphere of society, is it valuable? And I think if we're kind of left to our own devices, we will figure out, if you just think about the rare disease environment, 7,000 plus rare diseases, if you enable lots of different researchers to go looking for those targets, I'm sure over the next few decades we'll find solutions for a number of them. But is that where we want to go from a society perspective? I think that's the hard
if each
part.
drug costs $300,000 a year, that's a solution that is not a practical solution, right?
And the
How do you deal with
development
that?
cost is now up to about $2 billion per
billion
compound. That's
per
the
drug,
latest research you see coming out. It's not a cheap endeavor. So
Yeah.
I think it goes back to that original question: what do we actually want to try and answer?
Gary Gibbons, how do you grapple with that one?
Well, I guess I would cycle back to the example I gave. There is an importance, even in cholesterol. Part of the work of Brown and Goldstein was actually related to kids with familial hypercholesterolemia, a rare disorder that helped identify one of the molecular pathways for cholesterol metabolism. So I think of it not as an either or, but a both and balance that we have to maintain, that we gain insights into a broad target group of statins based on understanding that rare disorder. And now they're generic. And something where the cost effectiveness, I think, is quite compelling.
Right. Pennies a day for the statins that Brown and Goldstein sort of laid the groundwork for. These new drugs, though, $14,000 a year, and all of a sudden, then that is part of the value proposition, right? So, I mean, that's a difficult questions. Ron, did you want to address
let me
that?
offer a slightly different perspective, because I'm not a researcher, never have been, but I am an engineer. And so from an engineering perspective, when I hear the question, do you think that things will continue to be very, very expensive, or will they get cheaper? There's only one answer. Of course it's going to get cheaper. And because there is an observation, I think, that a lot of the techniques that we've been describing are technology-based and data-based, information-based. We've heard people talk about personalized medicine, about big data. These are capabilities that at the beginning are extremely expensive to develop. But the unit cost of production very quickly drops. The ability to search through vast quantities of information makes the unit cost very, very low. And then the promise becomes the elimination or the reduction of failures. That instead of 1 in 30 successes or 1 in 10 successes, you get it to 1 in 3 successes. And that changes the dynamics of the environment. Now, I'm speaking deliberately as an engineer, not as a researcher, so you can tell me that it doesn't work in this industry.
It certainly has not worked in biomedicine,
In many industries, the combination of the technology, and I'm pointing at this particularly because it seems to me when I talk to researchers, that research is getting so much more and more information-centric, technology-centric. And that's where potentially the promise lies, that this could be something that changes those success dynamics in this industry.
Yeah. Vince, what about this? I mean, one way you can imagine, and people talk about sometimes, is if you have better diagnostics that really pinpoint a disease, then you and say, okay, this is the mutation, this is the treatment. On the one hand, that's good because you don't have to give a drug to a huge number of people for whom it won't be effective. On the other hand, you'll have a smaller number of patients available to repay your $2 billion development cost. So, I mean, where do you see the technology
I think the good
leverage
news
here?
is that the cost of creating a diagnostic is a lot less costly than creating a drug. But you have to tie those two costs together in terms of a system, because you have to do that precision medicine together. Mm-hmm. And so I do think, and going back to what you were just saying, that you do drive the cost down, but there's a timing issue here because the costs are front-end loaded. So how you get those two things in alignment, I think, is one of the tricky parts of this. And so, because the benefits accrue to the patient over time, and you have the cost up front, whether it's a surgery or whether it's a drug. So I think we're going to have to get very inventive in terms of how we do those things.
Sudip, did you want to add something to that?
Yeah, the thing I'd add is just from the slightly different perspective of, in the 1950s, President Eisenhower had a heart attack while he was in office, and he got the best standard of care, bed rest. In 2005, my father, a textile worker in North Carolina, had a heart attack. He got $300,000 worth of care. On the other hand, his life has been extended by 20 years. And multiply that by 10 million people. And so there is this other piece of it, which is that you gain the value, the economic productivity of the people they're going to have a longer life. And so you have to balance those. And I agree. The fundamental question is how do you pay for it up front to get that end
Value
benefit?
over time.
Yeah, it's obviously well in industry. I guess you hope investors will give you money in exchange for return in the long run. I guess government's a little bit different because that's more not as clearly coupled together. You can you sort of have to rely on Congress to see the value of looking in the long term.
But there's a second piece of this, which is, how do you coordinate your care? And this comes back to you now have the opportunity with information systems going in the direction that they are to tackle some of these cost issues and reinvest in some of the technologies that we all know about. But when you think of 5% of the patients, costing, up to 50% of the healthcare system, there's a lot of room to redesign the system to create more room for innovation, in my opinion.
So what role should the government play in that? Anyone want to take on that question?
Well, one role, I think, from the standpoint of NIH and certainly NHLBI is related to that public-private partnership and breaking down the silos, even in terms of how we think about collecting data. We tend to go off and have a pristine clinical trial that, as you said, may be difficult to walk back into a clinical practice. What if we started to engage in more embedded research within systems that are actually functioning in the real world and collect that data now that a lot of that is digitized? And so I think it is going to challenge us to think about different models of how we learn new things about drugs or their additional benefits and make that part of our population laboratory. That's one opportunity we have with a digital age.
Yeah, no, I think that's a huge public education issue. I mean, I remember when your colleagues announced the latest iteration of precision medicine. One thing that they mentioned was, maybe your Fitbits or other electronics will be gathering data that then can be shared with researchers. And I thought, would I want that to happen without knowing? And then a little while later, I looked at my iPhone and realized it has been it's been counting my footsteps. I didn't ask it to. And, it could, but there's huge data there. Maybe, under what circumstances would it be appropriate for industry or researchers or somebody to tap into that? And, I guess it didn't turn out to be a question, did it? But somebody, if anyone would like to have a thought about that, I would be interested.
Well, certainly it challenges what we mean by the informed consent for research, and being sure that indeed we maintain those principles of beneficence and privacy and protection. But as you pointed out, there's data being collected on you right now that, and sometimes being sold, that you really probably weren't aware of.
Who knows? Maybe this conversation's being streamed across the world. Yeah. Oh, wait a second.
That could happen.
Well, Ron, let me turn back to your earlier comment about and thinking about how to make things more efficient. And one thing, as was mentioned, I am writing a book right now about biomedical research and the reproducibility issues involved in that. And part of it is, science is never going to be perfect. You're going to go down blind alleys and so on. But one thing that you could do, if you could figure out a way to do it, is you could reduce some of these easily preventable errors. People, using cell lines that are inappropriate, not authenticated or whatever, and so on. And the journals could play a very important role in that. And I wonder if you would say a few words about whether are the journals in general stepping up to this? Everyone wants to be published and sell, right? And so you have tremendous power to make a difference here.
So earlier on, I was hearing from Mary that you're writing a book on reproducibility, and now you've asked me a question about reproducibility. It seems to me that maybe we should be switching chairs. But let me take your point. So reproducibility is something actually which we in Elsevier spend a lot of time on internally, but also collaboratively with others. A couple of remarks, but I recognize that these are things that you will know very, very well. First of all, we think about reproducibility in two ways. First of all, is there enough information about the experiment to reproduce it in the first place? And then secondly, was the way in which the data presented ethically correct, or was it somehow doctored so it does not become reproducible? And those are two separate issues. They're both important, and we work on both of them. Within the individual environment of journal publications, there are some things that we can do. One of the things that we can do is make access to the underlying data easier for the reader. And so we've now connected papers to 200 separate institutional data repositories, so that if you're reading a paper, you can click a hypertext and you can go straight to the underlying data. These are very much pairwise links that we negotiate with each repository to make sure they're happy for this to happen, or the researcher's happy for this to happen. So that's one area that you can work on, getting the data more connected to the underlying research paper in the first place. Another thing that we do is there's a lot of work going on around methods, because one of the issues around reproducibility, and it came up about 9 months ago in the press, is that sometimes it's difficult to reproduce because the methods are quite complex. If you don't take all the steps the same way as the original researcher, you may not get the same results. It doesn't mean the original research was wrong. It may be you made a mistake. Maybe you didn't follow the steps.
Right.
Maybe you weren't presented with the steps correctly. So we've been working on that within Elsevier, and we've actually created a cross-science journal called Methods X, specifically to allow people to get into great detail about method. That's the second thing that we're doing. We're actually working with the NIH. Elsevier publishes Cell and the family of Cell journals, and Cell Press is working directly with NIH on issues around reproducibility, because you have a program going on over there. And we're working with an institution in Germany as well around, mostly from the second angle, the data integrity piece of reproducibility. So we see because the journal is meant to be the document of record, it should be the place where it has the integrity that you're looking for in reproducibility. And I think there's a lot we can do, especially now that we can move away from paper and we can go digital. We can help that environment move much more aggressively towards an environment where you have everything you need to know to reproduce the experiment.
That this is an opportunity as a community to respond to this challenge. And there has been a nice partnership between NIH and the journals as sort of safeguards of the public good of data. And I guess I would say that only add that we're instituting some of these things on the grant application process as well, such that we ensure that we're funding research that is based on rigorously done science, both in preliminary data as well as its execution.
Are you worried that this might undercut public support for science at some point?
Well, again, this is a public good, and it's critical for it to engender public trust and have integrity. And so as stewards, that's why we think it's an important thing to ensure that what we fund has that integrity, and we're setting a bar, if you will, of expectation of those who receive taxpayer funds, that this is the expectation of rigor, and coupled with the journal reinforcement of that, I think that's something the community will step up and do.
Yeah, and I must say that in my reporting so far in the book, I think that people are very upfront about this. They recognize it's an issue, and they're not trying to sweep it under the rug. And I think that's a laudable approach to it. But it's also, one has to be aware. Here we are talking about thinking about potential. Trying to convince Congress that this entire enterprise is heavily worth supporting, and this is an issue that can raise questions. Did you want to add something to that?
Sure. The example that you're seeing here is actually, you asked the question, what can government be doing to foster what's going on here? And that example is a singular example of NIH and industry working together. That framework can be provided by government, and part of that is the 21st Century Cures, part of it is other things that are providing that framework to allow that cross-collaboration, again, with a goal in mind. And I think that the goal isn't about a target for a cure, it's about that is one way of looking at reproducibility. How do we get promising candidates into the pipeline for orphan diseases? what kind of collaborations are needed for that? How does the engineering workforce need to be provided for by NIH in the same way that the molecular biology workforce is being provided for by NIH? These are things that require a framework and require funding. And when those two things are coupled, like they are in the 21st Century Cures Act, or when there are thoughtful appropriations as opposed to a continuing resolution for a full year, we can make a lot of progress. When those things are not done in a thoughtful way, we have that same organic, on a one-off basis, these collaborations, and that's a real problem.
Yeah, Vince, do you want to say, or
I completely agree with those points. And just, as you said, one of the questions you asked at the start about government and changing mindset and paradigm. I mean, I go back to the example you said about you're collecting steps and it's going into some database somewhere. I think the paradigm of research, the way we've been doing it, the single investigators, the multi-investigators, the randomized, double-blind, that all approach and the methodology piece which you were mentioning, I think a lot of that needs to evolve. We have massive data sets that don't really collect data in the same way but are equally very powerful as long as they're read appropriately and as long as they're reproducible. But that's definitely a paradigm change which we could embrace a lot of the technology pieces and actually use the power of that to do something different. So if you look at the development pathway for biotech or pharmaceuticals, it still very much follows that same paradigm which has been around for a long time. It's long, it's expensive, it's costly, it leads to a lot of expense at the other end. But I mean, there are ways to think about the requirements in a different way, more real world, more real time, and think about that paradigm differently. I think that's an opportunity now to follow up on that point.
Yeah.
I guess the question would be, if this is a problem, are we going to fund a program and divert some resources to getting at reproducibility? Because I don't think it happens on its own. And so when you're thinking about grants and what kind of grants are going to get approved, does this get into the queue in the appropriate way? I think it's been much more driven by, okay, this is really interesting science, as opposed to the methods that we need to be reproducible. We have the largest blood collection business in the world, and I can tell you when we look at our research customer base, by and large, they are not using the state-of-the-art methodologies in blood collection to preserve proteins and whatnot. There's a fraction of them that are. So I think it's a really interesting question that you started to investigate.
Yeah, and it seems as though there's a role for industry to come up with some solutions. It's partly. We could spend the rest of the time talking about my book, but we should probably. but we questions and answers are coming up momentarily. So let me just take one step back and ask Dr. Gibbons a broader question. We've been talking about technology here and these sorts of solutions to healthcare, but there are and moving things forward. But there are also, we're reminded that there are disparities in this country, around the world, of course, in people's health, and it's not purely a matter of diagnostics and drugs and treatments and all the rest of that stuff. There are other broader issues here, and I wonder I know it's something you've been thinking about a lot, and I wonder if you just want to say a couple of words, just sort of, just to broaden the conversation before Q&A here.
Yeah, no, I think that's another dimension of what we're talking about in terms of systems and data. It's appreciating not only that we may have your genome sequence, but in part related to digital age, we can get much more information about behavior and social context. And so if we're trying to address, say, asthma care and the NHLBI, yes, we'll have your genome sequence, but we also may have a sense of the neighborhood where you live and work or play, and the air pollution and ozone levels and particulate matter that may exist there that may also have an impact in your health outcomes, and have a way now of integrating some of that social context information. So similarly, in fact, some of the individuals at the lower end of socioeconomic status actually use these mobile technologies even more, because that's their real source to the internet. There may be opportunities then to put in reminders to promote greater adherence to their therapy by, again, leveraging some of these technologies. So we want to be sure that we don't create a digital divide. In fact, we try to leverage it in a way to maybe actually shrink health disparities. So I hope we take that inclusive approach.
That's very interesting. All right, I think it is time for question and answer, and I am guessing that there are going to be microphones circulating around in the room. Is that correct? So if you have a question, please raise your hand. Someone with a microphone will come to you. Please introduce yourself and ask a succinct question.
Okay. Thank you. My name is Mary Hendricks, and I have the privilege of serving on the board of Research America and also run a research institute for Northwestern University. I think I have a new chapter for your book, maybe something we could think about. As a working scientist, I think the public is unaware of the fact of the tremendous variability that exists in commercially purchased reagents that we all use in the laboratory. Many of these reagents come to us, they could be totally inactive, antibodies not working. It would be great if the government hosted a site for maybe preferred vendors or reagents, just a site where researchers could feed into it and say, you know, this one really doesn't work. Don't bother purchasing it. And it's just a thought, but it's something that we usually don't talk about.
Yeah, you think that would be popular, Vince?
As a manufacturer of very high quality reagents in New York, we'd be happy to be on the website as. So I think in the research community that might be popular.
Yeah. All right, thank you for that.
So you'd support that for the
Listen, we're very happy to get our quality out there in any way that we want, and you can, you know, in plenty of methodologies. I think that I don't know how broad that problem is. You're saying it's quite broad, but I know that you need the government to get involved with that one. To do that.
It is a conundrum, yeah, for sure.
I think you can create a website and start. There's a lot of websites that talk about these various things now.
There are. Although there's a website that lists the hundreds and hundreds of cell lines that are not authentic.
Yes.
And the journals, unfortunately, do not even go to them to see before, maybe Cell does, but many other journals do not go to actually see, and you can still see papers published that are based on faulty cell lines. So
that was part of your reproducibility effort that you were talking about before.
Yep. So, okay, anyway. I don't see where the next microphone is. Would somebody raise a hand? Who has a question? Here we go.
Thanks, Mary Woolley. So all the conversation about data, I thought was really important, and I wonder if we could hear a little bit more. Now, maybe from the perspective of, are we going to fulfill the promise to the patient of the highest and best use of data? We know from public opinion polls that we commission at Research America that very large majorities of the public say they would be willing to have the science community and the medical community use data to help assure better healthcare delivery, better health care for themselves and for other people. So your possible comments on how do we get there and what are the barriers that we might have to overcome?
Yeah, who wants it? You want to?
Yeah, just one thing related to Mary's point. We also made deal with this sort of challenge in history in which, as a researcher, we talk about research subjects, in which it was about my R01, in which you contributed to get my data that I'm then going to publish in Cell. What if we actually thought about it in terms of it being your data, and that you still have control over your data, and if you do want it shared, you might be able to change that equation about how and who shares it and under what circumstances. And to the degree to which a lot of the data and information we're talking about may actually be generated by you from your sensors or what have you, actually it is your data. And so I think this is one of the challenges for us. It's not necessarily going to be the PI who's going to be the sole determinant. It may, in fact, again, involve a different kind of partnership where the participants in research have a say about where their data goes.
I think it's a really interesting point. in the UK, a similar survey was done where people were much happier to release their data if they were told the data belonged to them as opposed to belonging to somebody else. And interestingly, there's a small company in the UK at the moment which is building off that and trying to create a mobile app where the individual on their cell phone own the data on the phone, and they're taking the cell phone to the clinician and saying, here's my data, as opposed to, you've got my data, I don't know what you're doing with it, and I show up and you tell me that I'm 35 years old and I'm actually 43 years old, or whatever it happens to be. So switching that over seems to be one of the ways, commonly now listening to you, that the discussion with the public moves away from what are you going to do, and what if it gets lost, what if it gets stolen, what happens to my health insurance, to it's your data.
And I think you also see it in areas, if you look at an example like patients like me, the amount of people who really want to be part of that whole process. I know the last time I spoke to them, there's tens of thousands of people who want their information to be used in a research type. They want to share their information so that it can be used once pooled together to enable further research in their specific disease area. So I think it's a really important point that given the choice, people actually want to be part of these things. And if you look at, I studied medicine, and the one piece that I learned a lot about in terms of cardiovascular disease was the Framingham site. I recently moved to Boston. I drove through Framingham, and this was a famous thing for me, and I nearly felt like rolling down the window of my car and asking people, how did it feel like to be part of this massive data set that really enables the cardiovascular research to move forward abounds. If we think about the power of Framingham or something like that on a global scale, where people are willingly sharing lots of things in real time, it is absolutely phenomenal what we potentially could do at this day and age with the technologies we have.
Yeah, Sudip, do you think that would actually encourage people to think more about, more positively about research in general, and feeling like if they're part and parcel of it, that this would be a sort of a good basically to have them a participant? And
absolutely, we have. There's anecdotes about that all around. So if you look at the Framingham study, there are people who are proud to be a part of the study.
Absolutely.
If you look at studies like the Black women study, another study where people are proud to be 20 years in, and the small certificate on the wall is worth a fortune to them. So that engagement is very important. The conundrum is that everyone feels this way, but if you've attended any of the sort of sessions about the Precision Medicine Initiative, you run into all these questions about how do you do this? How do you actually make this practical? Because everyone wants to share the data. There's a million veteran study. There's studies all around with lots of cohorts. But how do you actually blend that data together? And there's not an answer to that yet. And that is where this dichotomy between what the people in our citizenry actually want and what we're able to provide is taking place. And so the way around that is to have this, first of all, the discussion, which is what NIH has started. But then after that is the action to say, okay, what's it going to take? Does it take changes to privacy laws? Does it take giving a 21st century look at how we handle our data versus the 1990s HIPAA laws that we're looking at?
And we've done so well with the electronic medical records, haven't we? So which is the model for this, how not to do it. I think that we have another question over here.
Bill Haidt from Janssen. As a reformed academic now in the pharmaceutical industry, I want to make a comment and then ask the question. I really appreciated Dr. Gibbons' comments about curiosity-driven research and then the discussion about value. And I think one of the things that strikes me is to talk about the value of the generation of new knowledge and what that means to ultimately to society. But the question is, how do you frame that discussion of the value of generating knowledge as opposed to saying, well, we discovered a new target and therefore we're making a drug?
Was I supposed to answer that?
I think I was looking at you.
Well, that's what I think Research America does brilliantly, and the collective in this room. I think that's our challenge. That's the value proposition that has to be made to enable the public to have a grounded understanding of that importance of discovery. And some of it is, as you called yourself a reformed academic, this notion of actually how scientific discovery works, that they didn't know there was a statin at the end of starting that first question. And so some of that, as I said, by the nose has to happen as part of the process. So that's why I have difficulty with the either or. We all want the target. It's just, what's the best way to do it? And from the government standpoint, it makes more sense for us to do the research investment on that front end. Actually, quite frankly, the riskiest end, because there are going to be some rabbit holes. And it's helping the taxpayer understand that in the end, with the public partnership, there's going to be value. But the public part, it has to create this larger pool of public good where new knowledge is going to pay off at some point.
Yeah, we have 40 seconds left. Maybe one super quick question, although that's a wonderful point to end on, actually. I'm almost reluctant to take another question, but
particularly from this guy.
This is Victor Zhao from National Academy of Medicine, formerly the IOM. So the question which has been raised, 21st century cures certainly saying let's remove all the borders, let's move discovery faster to cures. But there is in fact a couple of issues we have to discuss. Gary mentioned human gene editing. As you know, in our academies, we're working very hard in addressing this issue. The question is, where should the responsible research, conduct research lie among the scientists, being able to know how fast to move, moving gene editing to human embryos, where the regulatory framework, and where the ethics around this. So how do you create the framework around this as we're struggling to think through this at the academies? But since you've been talking about really this issue about advancing science to faster cures, when technology is actually moving faster than, in fact, regulatory frameworks and others, and yet we say they're holding us behind, when yet there's, in fact, if you think of gene editing, there's a real reason to have that kind of oversight and framework. So can you say a word about this?
It's a fabulous question. I think we're already over time, and I wonder if maybe you could take that on over coffee, unless somebody has a 30-second answer. I mean, it's a really deep question, and I don't really want to give it short shrift. But unless somebody wants to take 30 seconds, I think we should leave that hanging in the room and something really interesting to talk about. So anyhow, thank you all very much for your attention.